Hello and welcome to Larkin Hospital.tv, the medical internet TV station. And today we're happy to bring to you Dr. Mendel, who will be talking about neurosurgery, again with another wonderful lecture. And so I'd like to begin by saying hello to Dr. Mendel.
Hey, Simon.
Welcome, and we're going to introduce the panel. Mohammed, would you like to introduce yourself?
My name is Mohammed. I'm MD doctor joining today from Toronto. Thanks for having me.
Thank you very much. And Dr. Bennett?
Yeah, hi, I'm John Bennett in Miami, Florida, ex-ER doc and lover of Hangouts. It's nice to be here with Richard. Thank you.
Thank you. And Lisa?
Hi, I'm from Bentonville, Arkansas. I'm a second-year medical student. Thank you.
Thank you. And Jesse?
Hi, I'm Jesse Contra, coming to you from Philadelphia, Pennsylvania. I'm a graduate student.
Thank you very much. My name is Simon Downs. I'm a second-year medical student, and I'm also a developmental psychologist in Tokyo, Japan. Okay, Dr. Mendel, please.
Okay. Well, you know, we started after the last session. We talked about maybe doing a feature on optogenetics, and that's what I am intending to do. Although yesterday, which is August 7th, this week's edition of Science was just released, and there were a bunch of articles in there pertaining to optogenetics and other tools, and other tools that are in use right now for neuroscience. So what I thought was I'd try to incorporate some of the new information that came out yesterday, and there's a great two-page overview called The Brain's Identity Crisis by Emily Underwood, and these are the referencing for the Science article. There's a really lot of material in here, so what I'm going to do is just kind of try to abbreviate some of it, and then I think I'll give you a good picture of why we are looking towards mRNA sequencing and optogenetics to find out a lot more about the brain. And I think it'll be obvious to you in a little bit what I'm getting at. So this, you've seen the reference slide. But here's the second slide. Does anybody know who this is? You've probably all heard of him, but his name is Ramon Cahal. It's Santiago Ramon Cahal. And Dr. Cahal is really credited as being the father of neuroscience. And Cahal was from Spain, a very rural area of Spain, like in the Pyrenees Mountains. And as early as 2005, a big international conference was held in Cahal's hometown to commemorate, you know, his fathering of neuroscience. And so what I'm going to show you is really, he thought that the neuroanatomy and neuroscience, he thought the most beautiful images were those of the retinal layer. And Dr. Bennett and I both went to the Medical College of Pennsylvania, and I can tell you, and this is pretty much what we thought of in 1989 as the retinal layer. And there's basically 5 different cell layers in the retina, and we talk about rods, cones, bipolar cells, amacrine cells, and you know, the rods and cones, they just respond to wavelength. So, you know, as early as 1989, we really didn't have that much knowledge of even the retina and its complexity. So again, here's Cahal, and like I said, he opined the retina was the most beautiful of all the cell types. Now, you have to remember that his imaging then was all just microscopy. That's all he had was microscopy. So a big issue in neuroscience is neuronal classification. It's really very, very complicated. For instance, interneurons have really come to light as the building blocks of all the brain circuits. Just like proteins, amino acids are to proteins, interneurons are to making a neural circuit work. In the brain, you'll have many neurotransmitters: acetylcholine, serotonin, glutamate, glycine. Glycine's really prominent in the spinal cord. Glycine is mostly an inhibitory type of circuit. But malfunctions of these interneurons are what plays a role in a lot of different diseases. And these include epilepsy, schizophrenia, autism. So the way that Cahal got very interested in neuroanatomy was one of the tools he used was invented by Camillo Golgi in 1872. It was a silver nitrate staining of cells. But, it's 1872, so he just happened to come upon silver nitrate for staining cells black. So it had no really specificity. All he knew was that it stained these cells, and he looked to see, what was staining and what was not staining. So Cahal started looking at all different layers of the brain, and he was very much a splitter rather than a lumper, right? And the splitters and lumpers, you'll see, they go to war in a short time. But he called all these different images the butterflies of the soul. That was his quote, butterflies of the soul. And he described a lot of the figures as stars, bird nests, baskets, spindles, and it was his opinion that these were the keys to higher cognition. So here it is in the 1970s, everyone thought that the retinal layer was really pretty simple, 5 cell layers, okay? As it turns out now, when I graduated in '89, that's what we thought, 5 cell layers. Now there's well over 60, and that's kind of a conservative estimate because we're looking at things through using different tools. So rather than just using morphology, shape, as the classification system for neurons, there's a lot of other things that come into play, and much of that has happened in the last 10 to 15 years. And the things that come about are first morphology, which goes back hundreds of years. Next is neurotransmitters. And so, the evolution of the neurotransmitters has really been all towards the latter part of the 20th century. Now, with mRNA sequencing and the manipulation using tools like optogenetics, we now know we can code neurons by their mRNA and know what their utility or derivation is. So here's another thing. Prions were a pretty hot topic when I was graduating from medical school. And these from Jakob-Creutzfeldt disease and scabies and that kind of thing. So I never really thought of prions as in anything outside the context of things like Jakob-Creutzfeldt or however you want to pronounce it. But what this next slide shows is kind of a model of how diseases like Alzheimer's disease and also Parkinson's disease, they lead to accretions of cellular debris, and that was big in 1989. The talk about with Alzheimer's disease of beta-pleated sheets and alpha sheets, and I can remember in '89 people were thinking, oh well, there's some correlation between aluminum foil, eating food wrapped in aluminum foil and Alzheimer's disease. Is very, very, very primitive. But what we've learned is that in Alzheimer's disease, with the buildup of seeded areas and accretions into cells, you start a process where you get intracellular occlusion bodies, and you get them in Parkinsonism too. And there are many derivations for the genetics of Parkinsonism, and most are not hereditary. They're more sporadic. And so, we would listen to the neurologist describe different types of dementia, as frontotemporal, entorhinal region, and that was really confusing, especially to a surgeon. But we didn't. What we were able to do was find patterns that seemed to correlate with where the inclusion bodies were in different cells. So, for instance, with Lewy body disease, which you see more in Parkinsonism, there's actually an anatomic pattern to where the Lewy bodies begin and how they, where their predilection is to build up in the brain, and that is what causes the different appearance of different types of dementia. We used to just refer to frontotemporal dementia mostly as Pick's disease, but we found, there's a lot more than just Pick's disease. And we found that by the use of things like optogenetics and now the mass spectrometry and large screening of the ability to screen big, 10,000 cells at once and find all their, their mRNA chains. This is the appearance of different neurons in kind of what Dr. Cahal called the butterflies, butterflies of the soul. Now, recently, and some of this appears in the Science edition from yesterday, this geneticist, Steve McConnell, this is just notes so I could remember to tell you about it. Steve McConnell and the group at Harvard developed a technique called DROP-seq, S-E-Q. You see it here, DROP-SEQ. And what that could do is profile messenger RNA in thousands of individual brain cells at once. And then this fellow, Sten Lennerson from the Karolinska Institute in Sweden, has a little bit slower technique, but he could do 3,000 individual cells at once. And he says it's very cost effective, and he feels that, quote, every little corner of the brain can be typed in, in the very near future. So this is a slide. Now, I didn't get permission, so I didn't print the whole slide. But the blue area, green area, and red areas represent different patterns of like, for instance, the green is you'll see the entorhinal involvement, and then the involvement of the fronto and temporal lobes. This is more of an Alzheimer's disease model with inclusion bodies. This, however, is the Lewy body inclusions, which do seem to begin in the brainstem. So in a short time, you're going to have a much better anatomic idea about the pathways, the similarities and differences between Alzheimer's, Parkinsonianism, and other causes of dementia that we just didn't have much insight to until now, because everything was really based on morphologies or on clinical presentations where you required a neurologist took a long time to get the subtleties of where dementia arose from or what the mechanism of action was. I was really impressed when I saw a neurologist who had given a diagnosis of Lewy body inclusion disease. This is in the late 90s. Yeah, no, maybe 96 at Jefferson. It was very good, and I thought it was a very sensitive exam where he had the gestalt of where everything had begun and where the errors in higher thinking came. So in 1975, the Nobel Prize got awarded to a former Swarthmore student who really came up with the innovation of reverse transcriptase and the way in which RNA worked. And my mother, she was really interested in this, and I remember her telling me about the Nobel Prize, how big a deal it was, and I was 14 then, and I was having none of it really, but I do still remember her telling me about it. My big concern in 1975 was a boxing match between Cyclone Hart and Benny Briscoe. I think it was on November 18th. That was the big thing that happened in 1975. But I do remember my mom was appalled that I didn't take more of an interest in the mRNA. But it turns out that is actually really what's allowing us to get more information about neuronal typing. So what happened about 10 years ago, I think it was 2005, a big issue in neuroscience is the fact that we don't have a good way to classify different neurons, and between different scientists there's so many overlaps of what one person was calling one thing or another that in 2005 this fellow Giorgio Ascoli, who's at George Mason University, he termed the meeting the Tower of Babel meeting, and that's the meeting that took place in Cahal's hometown. And people from all over the world in neuroscience were there to attend that, and within a short time they realized that it was, quote, a fool's errand, and that here we were, we thought there were 5 cell types in the retinal layer, when in actuality there's at least 60. And morphology had given way to neurotransmitters like we talked about, acetylcholine and serotonin. And then that's given way to what some people have claimed is the most promising marker of cellular identity, and that's gene activity, or as Maslin says, RNA sequencing. He says it's probably the ultimate way to classify all these cells. So in a very short time, we're going to see much more discrete classifications of cells. I think it'll get incredibly complex pretty quickly. This is the view of the quote-unquote relatively simple retinal idea that turned out to be not at all simple. And like I said, there's over 60 different cell types now that are because of the permutation of not just being the morphology, the shape of the cell, but also you add the permutations of the neurotransmitters, and then the permutation of their gene encoding. So the gene encoding is really important now because that's how people, like the fellow at Stanford, and I'm going to show you a video of him next week, that's how people began to manipulate the cells, the genetics of with naturally occurring opsins and that kind of thing. They're called rhodopsin channels, so that optogenetics could be used to identify functional areas of the cell by turning on and off different neurons. And that was really the first time where we had a tool to use on a neuron or on the brain cell where we could turn different things on and off. Because if you think about how we've stimulated the brain for the last 100 years, right up until now with deep brain stimulation, it's been with electrodes. And it's really an all or none phenomenon. You're not selectively stimulating different channels. You're stimulating everything. It's very much like ECT. It's not selective. It's not, a sensitive process at all. All right, so this again was the idea that maybe Alzheimer's disease, maybe Parkinson's disease isn't as different from some of the prion-related diseases as we think. And we're going to get into that more when we get more into the optogenetics next week. But really, this is a kind of a big change in thought. Not the technology, the idea of going from morphology to then synapses and transmitters. That's how we've progressed. But the idea that now we can selectively turn on and off different neural channels and things like rhodopsins are going to just add tremendously to the toolkit or the armamentarium of figuring out what things do, what things do specifically, and then we're going to have a lot more in the way of knowledge, not only structural knowledge, not transmitter knowledge, but you're going to know the functional purpose of that cell, and you're also going to be pretty much given a pathway to the origin of that cell, because you can just look back and see how the cell was derived. So this is really going to open up a tremendous amount of scientific activity over the next 10 to 15 years. And I think when we get to the optogenetics, and I have that talk ready for you next week, you're going to be amazed at how specific things have gotten in just the last 10 or 15 years. It's really quite a long way from where I ever imagined things were going to go in 1989. Are there any questions I can answer for you at the moment?
Well, thank you very much, Dr. Mendel. It's a very, very interesting lecture, understanding how light is used to control neurons, which have been genetically sensitized to light, and how using optics and genetics we can come to control and monitor the activities of individual neurons and living tissues and understand more things such as Parkinson's and Alzheimer's, and leading to better classifications of neurons, gene encoding. It's really amazing. I was thinking about it, and one thing that came to mind is that you were talking finally about how this will help us look back into how cells were derived, and to help us understand the interrelations between these diseases, and how Alzheimer's is resembling Parkinson's, and made me think about how looking at our universe and looking at the origins of our universe is helping us understand our present state, the present state of our Earth. So I'd like to ask the panel if there are any questions.
Yes, I have one, Simon. And thank you, Richard, for a fine presentation as usual. I guess I was surprised you said anatomically you can distinguish between the different types of dementia. Is that correct, Richard?
Well, I would like to say that I could, but I'm a neurosurgeon. A very good neurologist can really track down the subtleties of where the dementia originates from.
Right.
As I said, I'm a surgeon. I'm not that good with that I can confidently trace the origins of the dementia, but really they're explained so much better. I'm going to get permission this week to use the slide, and then I'll give you the patterns of how this develops. So basically, this is the AD, Alzheimer's disease. This is PD, and this is this may be a multi-systems atrophy. I'm not sure, but yeah, I think with the use of MRA, you're really going to be able to find the origins of a cell right back to before stem cells, and I think I think the ways we decide about dementia are pretty in some ways crude, with the Parkinson's disease, which we label a movement disorder, and Alzheimer's disease, which we call a dementia. We see a lot of Parkinsonian patients that do develop dementias, and that's typically been classified into things called like Parkinson's plus syndromes, which include diffuse Lewy body disease. And what we're really seeing isn't, we're not seeing, maybe in the 70s or so, you would learn neuroanatomy as though they were structures. And we know now that most of what we're interested in isn't one structure. There isn't one elegant place for the function. It's, speech, understanding speech. They're loops, they're circuits. So what we're going to find is that there's dysfunctions within circuits, and I'm sure there's a myriad of ways in which these circuits overlie each other, and that's what we're going to learn about. I mean, we're going to get, I think, a lot more insight into what's going to be looked back at as, relatively crude classifications compared to what we're going to see in the near future.
Richard, that kind of leads to my next question. I wonder if it's really clinically important to distinguish what type of dementia as far as treatment. I suppose some types of dementia, if it's Parkinsonism, I don't know if it would be affected by L-DOPA, if that L-DOPA actually improves the dementia or really helps the motor types of problems with Parkinson's. And what I really want to know is, have you ever been asked to do a biopsy by a neurologist that really wants to, that goes to you, hey, Richard, I really want to know what's going on. I want to see what type of dementia. We do a biopsy. Does that happen at all in neurosurgery?
I have not heard of that for dementias unless there's concern about an infectious etiology, like we would do in the 90s with AIDS patients and trying to determine whether it was, CMV or abscess. But when it's a dementia that may be the result of infectious disease, you might be asked. But with dementias, there's a lot of different dementias, and many of them are reversible. Okay. And so you always want to think back to medical school and the Wernicke's and Korsakoff syndromes and folate deficiencies, that kind of thing. There are a lot of things that are reversible, so you don't want to lose sight of that. Not everything is an irreversible process, and we're going to maybe understand things better so that maybe the issues. I'm thinking mostly of the interneurons. If you have an interneuron problem, maybe there's a specific mechanism that some may be solvable. Maybe they won't, but you know, optimistic about it at least. The idea, though, that now we're able to discern function in different receptors on the neurons, really pretty impressive, though. I mean, we really went from being pretty crude, crude 50 years ago to being, we're working at a subcellular or subneuronal level now.
No, Dr. Richard, brings that important point. I'm really surprised with all the advanced technology and the techniques they're trying to use. I mean, back in 2006, I participated in neuroanatomy research where we would be injecting dye and then let the dye stay for about 10 days and then come and slice the brain to look into those projections. I guess now they're looking more into the interneuron, how it functions, and that's amazing to see.
Yeah, I think I was thinking about this recently. I was at the University of South Florida recently, and they had this amazing virtual lab, and they even have a anatomage. It's like a digital large table, like a gurney, and they dissect what appears to be a cadaver, and it's all pixel dissection. You could feed CTs, MRIs in it. It's really amazing. But I'll tell you that one of the best things that I had was I think that with managed care, I'm talking about the late 70s, the early 80s, with managed care, autopsies kind of stopped being done. Insurance companies didn't want to pay for services on dead people. Okay, so that was bad in a lot of ways because you really learned a lot. You didn't have as much time learning anatomy because you weren't doing a lot of autopsies, and the pathologists especially weren't. But when I was in London, I spent a lot of time with a neuropathologist over there because they still did tons of autopsies, and I must have cut more brains over there than I did, for years here. And so it's really to hold the brain in your hand and look at it in 3 dimensions. You have a much better gestalt for what's going on. I think you may be able to learn gross anatomy if you're not going to be a surgeon by doing it on a digital mannequin, but I think that 3-dimensional input's important. But neurosurgery, you really have to know where you are in the brain, and you've got to be so familiar that I think it's a shame that people don't get to cut the brain as much as we used to. Let's see.
Richard, can I ask a question?
Yeah.
You said a lot of dementias are reversible. I'm surprised at that.
Well, think about an alcoholic, okay, who may be on a binge for several days and hasn't eaten, okay, and they're just drinking, okay. You learn this in your first year or two, second year of medical school, maybe. They can have a folate deficiency and get into a Korsakoff psychosis that that is reversible for a short period of time if you get them treated. You know how they were treated, like in the 50s? Folate and thymine were added to the pretzels that were served in bars. That was an easy way to get alcoholics their thymine and folate, so they didn't have, Korsakoff syndrome, and that seemed to be an acceptable way to treat them.
Okay.
You know, there are things, you do have to keep that in mind. Even with peripheral neuropathies, you have things that are reversible, that aren't reversible. So that's something you want to always keep in mind.
Okay.
One thing you always worry about is a young person with especially a young person, big change in mental status. You've got to think of herpes encephalitis, because if they don't get treated in time, you can get permanent neurologic deficits that are devastating. So that's something you always want to keep. As an emergency room doctor or whatever, you'll always keep a herpes encephalitis in mind when you're seeing a patient with a change in mental status, because if you're too late in initiating treatment, you really expose them to the risk of a permanent neurologic deficit, higher cortical function.
To diagnose the infectious, I guess that's really not up to you. They come to you after it's diagnosed, and if they have a
but if you're seeing somebody acutely and their history is that consistent with a herpes encephalitis, you're going to expect that they've had their acyclovir hung already to arrest that. I mean, you don't need a neurosurgeon to tell you to hang the acyclovir. You just got to keep in mind that that's on your differential of things that need to be treated urgently with a change in mental status. You don't need to wait for an LP. You don't need to. I mean, you just hang the acyclovir and keep rolling until you figure out what the problem is. Because sadly, if you wait too long, you wait 8 or 12 hours, you'll see both temporal lobes lighting up, and that's not a good thing.
Okay.
Okay.
Jesse or Lisa, any questions at all?
No questions.
The reason I wanted you to see this is it kind of gives you an idea of how the technology marches forward and how we get from morphology type of things under a microscope, shapes. But these are the tools that now we're using to get to function and purpose and derivation. It's very, very interesting. When you look at oncology, one of the hottest topics in oncology over the last decade has been the input of the in immunology, and that's now being hailed as really like a fifth weapon against the development of cancer. So I think you want to kind of look at the tools that are being amassed to treat whatever you're going to treat, because
I think Richard accidentally puts the mute button. Richard, you're muted. Oh, I muted him? Okay.
Dr. Bennett, it says that you muted him.
Oh, I did? Oh, well, I did. Well, let me check here.
While you're working on that, I have an observation. When you're talking about looking at the activity of cells and the gene expression, and that tells you a lot about that, it made me think about someone said years ago that if you just knew what a person spent their money on, then that's a good enough picture of what activities they're involved in. And so I thought about the department stores that offer like discount cards or something that they have you fill out, and then you're supposed to hand them that card every time you make a purchase, and then eventually they've got this picture of you based upon what all you have purchased in their store. And so then that it's just creating a profile of somebody. And there can be multiple ways of using that. But anyway, it just kind of goes back to this very similar idea that by using the gene sequencing and looking at what is this cell involved in doing, it's got to make certain proteins to do certain activities. So it's basically what the cell, it's not buying, but it's making, but it's kind of on that same track, if you all get my drift. So very interesting.
For sure, Lisa. Yeah.
Right.
Okay, we're having.
All right. Thank you very much. Having some technical difficulties here, but I think this is a good time to bring our meeting to a close. I'd like to thank Dr. Mendel for his wonderful presentation and a very unique, special introduction into optogenetics. Very exciting, and we look forward to understanding this better in the future. So I'd like to thank everybody else, and we will see you next time.
Thanks, everyone.
Thank you, Simon. Thanks everybody for coming.
Thank you.
Thank you.